To think about protein structure and understand it is looking at the sickle cell anemia example. Essentially what happens is that normal blood cells are in a disc shape like this, but because of a mutation in a particular gene, your cells become this blood cells become this crescent shape. And what that results in is the clumping of your blood cells. And because they clump together, blood flow is prevented. And because hemoglobin carries oxygen, cells don't get enough oxygen, and oxygen is required to make energy, so it causes a lot of complications. What happens is, because we're changing the primary structure of the amino acid, which refers to the sequence of amino acids in a polypeptide chain, and because we're changing that sequence from having this glutamic acid over here to the valine over here, we're going to change the way in which the overall protein is going to fold. And that's because the R groups of these amino acids are going to be different, right? And because the R groups are different, they have different chemical properties. So the R group of valine will not interact with the neighboring amino acids the same way as the R group of glutamic acid. And that will result in a different folding of the protein. And so the overall functional tertiary structure of the hemoglobin will change. So the tertiary structure is the overall three-dimensional shape. So the overall three-dimensional shape we get here might look like this, like a normal hemoglobin protein, but here you get that crescent shape because we're changing that primary structure. That has a result in the subsequent secondary and therefore tertiary structure. So secondary refers to like these formation of alpha helices and structures called beta pleated sheets through hydrogen bonds, and then tertiary structure is like how the everything just like interacts with each other and gives your overall shape. As the cells become more crescent shaped, they begin sticking to each other and they begin clumping and they prevent oxygen from being transported. It's because of the mutation in your DNA, which alters the primary structure. As we can see, the A is being replaced by a T, and that's going to result in this disease.